Everybody, it's Mr. Matthew here again for AP Biology Essential Knowledge 3C1 video. And in this video, we're going to talk about how changes in genotype can result in changes in phenotype. And just as a quick review, genotype is going to be how we define the genetic makeup of an organism, and the phenotype is what it physically looks like. So here we go. So one of the things that we're going to start with is to talk about the concept of mutations. So we need to be able to predict how changes in genotype may be expressed as a phenotype provides a variation that can be subject to natural selection. So again, first we need to define the concept of a mutation. And so what I have here is I have our first example, which is no mutation. And this no mutation is going to be that if the DNA is TTC, we will produce AAG on the mRNA level. AAG will then be translated into lysine. And so this is our no mutation wild type example. And then what I have is I have three different changes on the DNA level. One that we're calling a silent mutation, one that we're calling a nonsense mutation, and two that we're referring to as missense mutations. And so what I want you to do is I want you to pause and think, and for each predict what kind of protein change we're going to see if we have a silent, a nonsense, or a missense mutation. So pause and think. All right, so hopefully you remembered back to your earlier biology day that a silent mutation is one that is a mutation because it's a change on the DNA level, but it is silent because there is some redundancy within the genetic code at the codon level. And so if you were to change TTC to TTT, you end up still producing the same amino acid because both AAG as a codon and AAA produce the same amino acid. And so on the protein level, there would be no change for this. Again, if you don't have a protein code or a codon key, this is, you're just making some guesswork on here, but with a codon key, this would be a little bit more straightforward. All right, so what about a nonsense mutation? So we have ATC going to UAG. And so you might have been like, wait a minute, UAG, I know what UAG is. UAG is one of our stop codons. And so now what's going to happen is this is called a nonsense mutation because rather than having the long amino acid chain that has lysine in the middle of it, instead we're going to drop a stop right in the middle. So this is going to produce a truncated or a not full protein. And generally speaking, this is a nonsense mutation because we're not going to have the amino acid sequence that can fold up to produce the correct protein. That leaves us our last two examples that are missense mutations. We have what are known as conservative and non-conservative. In the case of the conservative, what we see is that we're going to mutate lysine for arginine. Both lysine and arginine have similar properties. They're going to both be basic side chains, and as a result of them, they're going to have very similar properties as a result. And so now when we look at the impact on folding and we look at protein shape, it's possible that this missense may not have a dramatic effect on the protein. It will depend on what domain this is in the folding and how crucial it is to the hydrogen bonds or maybe even some of the other structures involved in the protein. But it's possible that you can get a conservative mutation and still produce a protein that is effective. In terms of a non-conservative, this is where we're trading out one type for a very different type of chemical property. In this particular case, this threonine is going to have a very different chemical charge and chemical properties than our original lysine. It's not going to have that charge like we originally saw. And so it's just going to have a very different chemical set of properties. And so therefore the folding and the tertiary and quaternary structure, possibly even the secondary structure, are probably going to be affected, and you're going to end up getting a different overall shape protein. It will again depend on where in this protein this is. If this is in one of the regions that is a transmembrane region, or it's a key pocket or fold, this is going to have a profound impact, and we're not going to get a protein that's anything like what was originally made. And also, if we were to switch out say a polar for a non-polar, we're also going to get very different reactions when those amino acid chains fold up to try to form the protein. All right, so now let's look at another specific example. And so now we're going to look at the specific example of hemoglobin, and I've got two forms of hemoglobin. And for this, I want to create a visual representation to illustrate how the change in the DNA nucleotide sequence can result in the changes in that polypeptide. And so here, what I actually see is the chain of amino acids for 8 of the key amino acids in hemoglobin A versus our hemoglobin S, which is our sickle cell variant. And so what we look at here is that we see that right here in the 6th position, we trade a glutamine for a valine in our sickle cell version. And so now the question is, what does this mean? How does this end up affecting our overall polypeptide that is produced? And so what we're going to do is we're going to need to look at our amino acid chart, and we're going to want to identify what the properties are of these two amino acids. And what I see is that my glutamine up here in the upper right is one of our electrically charged side chains. And in fact, it's a negatively charged side chain. But our valine is one of our non-polar side chains. So literally, these are going to have totally different side chain properties. The glutamine is going to want to ball into a hydrophobic core, whereas our valine is going to be looking to make, maybe a salt cross bridge or something like that. So as a result of this, this protein is going to fold completely different if you substitute out a glutamine for a valine. And glutamine is the more common form. The hemoglobin A is the more common of the alleles that we see in the population, as opposed to the sickle cell hemoglobin. And so hopefully this allows you to visualize how when those proteins fold up, the folding properties are going to be quite different, because we end up seeing that the glutamine is no longer going to be able to make those salt cross bridges, or not going to be able to have the same properties as we're accustomed to seeing. So now let's explain this connection between the genetic variation we saw in those organisms and phenotypic variations in the population. And so here's a classic diagram that we see where we can compare the distribution of malaria over here on the left to the phenotypic frequencies of what is known as sickle cell trait. And so what we'll see is, hopefully you immediately look and say, wow, there seems to be a pretty strong correlation. Now, obviously not everywhere that we see malaria are we seeing the sickle cell properties, but we are seeing there's a commonality in that pattern that we're seeing in these two areas, that where you find malaria, you're seeing sickle cell. And this led to investigators looking to see, is there any sort of advantage to having sickle cell trait? And it turns out, as we now know, individuals that are heterozygous have what is known as a heterozygous advantage in a malarial-rich environment. In other words, if you are born into an area where malaria is endemic and there's a high amount of malaria, if you have sickle cell trait or you are heterozygous, you are going to have a much lower ability to contract malaria. You are going to be resistant to malaria. Now, you are more prone to having some of the symptoms of sickle cell trait, but the trade-off of having slightly reduced oxygen transport and slight increase in clotting is offset by the deadliness of malaria. And so heterozygotes in this area have what is known as heterozygote advantage. And so as a result, there's selective pressure favoring those who are born heterozygous. As a result, we see that in areas where malaria is endemic, you will find as high as 20% sickle cell trait in certain regions, because there's selective pressure on those populations to survive in those areas with malaria. All right, so I hope that was a helpful review of both our mutations and how genotypes affect phenotypes, and how we can connect that back to evolution. And I will talk to everybody soon.